and good day, everyone. It's wonderful to see you and wonderful to have you here for this very important discussion and for a focus on the outstanding research that is being done in the Northeastern College of Science. You may know that brain injury, brain disorders are a huge public health issue. They may be a family issue for you. For example, in 1 year in the United States, almost 20% of the population receives some treatment for a mental health disorder. A significant number of people, very significant, are treated for brain injury. The focus on maintaining brain health, on restoring brain health, is one of the great challenges of our time. At Northeastern, in the College of Science and across the university, we have multiple distinguished investigators who are putting their minds and their research efforts towards solving the problems of brain health and brain disorders. And it's my honor today to have two of the very top investigators at Northeastern with us today to speak about their scholarship, to address the questions of brain health, and to discuss with you the unique approaches that they are taking to meet these challenges. We will have a format that will be friendly, conversational, and welcoming. And I'm saying this because I want you all to understand that you can put any question you like in the chat. There is no stupid question. I teach my trainees that all the time and my students. There is no stupid question. If you want to know something, please don't hesitate. Put it in the chat and we will be happy to discuss that. So what we are going to do is I'm going to introduce our speakers in a moment, and then we're going to have a short presentation by each of them where they'll talk about their particular approaches to brain disorders and to especially using imaging approaches to brain disorders. And then we're going to open this up for a discussion. So we have a sort of a panel discussion format today, and I look forward very much to hearing from each of our distinguished speakers. Professor Craig Ferris is a professor of psychology and pharmaceutical science at Northeastern. He trained at New York Medical College in electrophysiology, and he also got training as a postdoctoral fellow at Harvard Medical School in physiology, neuroendocrinology, and peptide chemistry. He was formerly at the University of Massachusetts Medical School and became interested in functional MRI. He has a distinguished history in behavioral neuroscience and neural imaging, and we were very honored to have him join the faculty of Northeastern. His interests are in early brain injury and the possibility of neurodegenerative brain disorders that occur with aging. Professor Sue Whitfield-Gabrieli is a professor of psychology at Northeastern and founding director of the Northeastern University Biomedical Imaging Center, NUBIC, here at the university. And again, we are deeply honored to have her on our faculty. She received her training in biophysics and physics, a PhD in mathematics, and another PhD in psychology and neuroscience at the University of California, Berkeley. She has held research scientist positions at Stanford and MIT, and we were really fortunate to attract her to Northeastern and to count her amongst our distinguished faculty at this point. Her goal is to understand the basis of psychiatric disorders and to promote their translation of this knowledge into clinical practice. She uses neuroimaging techniques to investigate the underpinnings of development as well as pathophysiology of psychiatric disorders. And she is going to speak to us today about some of her approaches to mitigate these disorders. So without further ado, let me open to Professor Ferris and Professor Whitfield-Gabrieli the opportunity for you to hear about their research and to hear how in the Northeastern College of Science, they are moving the needle on addressing devastating brain disorders. So I'm going to turn over to Diana, who's going to run a few slides, and to Craig Ferris to begin.
All right. So Diana, just hold off for a moment. Hello, everyone. Thank you, Hazel, for the introduction. I've been at Northeastern now for 14 years. Prior to this, I was at UMass Medical School for 28 years. And so the question would be, why did I move from the medical school, which is in Worcester, down the street to Boston, to Northeastern? And the answer is the College of Science built us a wonderful imaging center called the Center for Translational Neuroimaging. So what do we do at the CTNI? We do magnetic resonance imaging on awake animals. And for many of you, I'm certain you probably are familiar with MRI or have had one. And essentially, you're asked to get into the magnet, and you're the source of the energy. It's totally non-invasive. You're in the magnet, and they'll take pictures. The only thing they ask of you is to sit still, because if you move, you can't make the pictures. Well, we do the same thing. We do them with animals. And while we can't ask our animals just to sit still while they're having the picture taken, we've developed all of this technology for imaging while they're fully awake. So with that simple background, what are the kind of studies that we do at the CTNI? And while we do many, okay, we do work in Parkinson's and Alzheimer's and neurodegenerative diseases and head injury, which I'll talk about, and drugs of abuse. The one that I'm going to share with you today is this whole idea about mild head impacts and how they affect acutely and in the long-term brain function. So on that, Diana, can I have the first slide? Excellent. Okay, so you're all familiar with this. And it's called the bump on the head, the serious consequences of repetitive mild head impacts. And here's just examples in just collegiate sports of all of the type of, or just the frequency of these kind of head impacts. Now, I'm a granddad with 8 grandchildren, and my oldest grandchildren are all playing sports right now. They're playing lacrosse and they're playing soccer. And they've gone to great efforts at the Pee-wee level, this junior level, of mitigating the incident of head injury. You know, in soccer, they're not allowed to head the ball. And so forth and so on. But as you move up into high school, and certainly as you move up into college, things can get very, very bad. And so this is just an example, but it's not only here in sports that we have these problems with these head impacts. Can I have the next slide, Diana? But these are part of our everyday life. And here's an example I'm giving. Bicycles, told years ago to wear a helmet. It's a good point. The elderly falling down, car accidents, and in the military, active military. All of these things can relate to what would be mild head impacts, head injuries, with the long-term consequences of increasing risks for Alzheimer's, Parkinson's, and dementia. Can I have the next one, Diana? Okay, so I have this, the National Football League got it right. And I don't know if you were fans of football, but if you watched it this weekend, and I'm sorry if you're a New York Giant fan, but your starting quarterback, Daniel Jones, had a concussion. He had an impact, and they took him off the field, and you will not see him for 2 or 3 weeks. So he's under a concussion protocol that they just don't want to subject him to another hit. So here we've taken the lesson from the National Football League literally at the College of Science, at the CTNI, and we took their models that they use to study head impacts in their football players, and we translated the physics of those models and the engineering of those models to our own so that we could actually reproduce those in animals, particularly mice and rats. And so we developed on the end over here, I don't know if you can see my pointer, you probably can't, but novel animal models of head injury that reflect those that are mild and that were reported in the National Football League. In the development of these novel models that reflect the human condition, okay, we've also developed technology, imaging technology that's cutting edge, so much so that we've had 3 patents, all of which have been issued to Northeastern University on our technology that allows us, and I'm going to show you examples in just a moment, allows us to actually look at subtle changes in the microvasculature, capillaries, and their flow and the relation to the neurovascular unit. And so these patent technologies are exciting in of themselves, but with these technologies, there's also been a spin-off company called Imaginostics, which is actually taking this technology and getting it into the clinic, which is incredibly important. So all of these things have been developed in preclinical models, and they're finding their way to the clinic. We do a lot of work with pharmaceutical companies. Now that they know about these models, we have 2 companies that have drugs in trials, looking at our model to see if their drugs can enhance recovery from these mild impacts. And probably the most exciting thing is my move from UMass Medical for all those years to Northeastern is my interaction with undergraduate students and their research and their publications. The Center for Translational Neuroscience, we train anywhere from 12 to 14 undergrad students each year. Not each year, each semester. So there's probably close to 40 to 50 over the course of the year. And these students come into the laboratory. They're not physicists or engineers. Actually, most of them come from a neuroscience background, behavioral psychology, so forth and so on. And we train them in imaging. We train them in data analysis. They design their own studies. And these are hypothesis-driven studies. And these students then will publish these as first authors. We send them off to international meetings. Most of those international meetings are being held in the United States. I haven't shipped anybody overseas yet. And so it's a wonderful opportunity for the graduate students to actually do research, publish papers, and give an opportunity to give talks. And I would suggest to the moms and dads out there that have daughters and sons attending Northeastern, they may not be physicists or engineers, but I would certainly encourage them to come and visit the CTNI and see what we do and how we do it, and maybe talk science. All they need is an inquiring mind and an ambition to get into what we do. Can I have the next slide, please? So here's an example of what I call the mild head impact, with the point there, more than one is too many, and this is the case for the NFL. If they take you off the field, you're not going to see you in the immediate future. So it's the bump on the head, and all of you have probably had this. When I played sports as a young person, I would be trashed out on a field someplace, and I would crawl off the field, and it was always, well, rub it, shake it off. Let's rub that and get back out on the field. That was pretty much what we were told when we were young. Here's an example of this. So here's a healthy, normal animal, and this white area that you see it's called the impact site. All that is the edema on the overlying skull from the connective tissue and the skin. So that's the bump on the head. Here's a normal animal. You don't see anything like that. So now we're going to do the study where we have an animal. They're awake animals. We're going to bop them on the head. And that's my euphemism for the hit on the head, the impact. We're going to impact them once. All right. And so you're looking at data from a paper that was just actually published in the last 3 weeks, where we're looking at 1 impact, 2 impacts, and 3 impacts, and their effect on small blood vessels, the microcirculation, the capillaries. And with a single impact, and look at the red area. The red area is just showing you an area that has changed its permeability. This is an area that we've affected blood flow and the permeability of the blood-brain barrier in this area. And so with a single impact, and the impact is always dorsal, it's on the top of the head here, you see that these red areas are areas that have been affected. Okay, so that's with 1 impact. With 2 impacts, you see that it's even much greater. So the 2 impacts is the next day, we bop them on the head again, and we're seeing that we're covering a much greater area. But if we go, and on the 3rd day, give them a 3rd impact, this is the problem. It's almost global. It's almost over the entire brain. Now, this is the immediate acute effects of these things. And you might ask yourself, well, do they go away? And the answer to that, 1 impact does go away. But if there's 2 impacts in this short period of time, these repetitive insults, the injury does not go away. And so here we have is neuropathology. So we've actually taken brains from animals that have been only hit twice with these mild impacts that cause the bump on the head with no brain damage. There's no crushing of skulls or anything else like this. just the bump on the head. If we do it twice, and we look at these animals 3 weeks later, which is actually a couple of years in a human life, and we go to a certain part of the brain, which is the substantia nigra. Don't expect you to know this, but this area of the part of the brain is an area that contains your dopaminergic neurons, which are very, very important in Parkinson's disease. The loss of these neurons results in Parkinson's disease. And what we look at this, the control animals, all of this green fluorescence that you see over here in this part of the brain that houses our dopamine neurons. What you're seeing here is animals that are sham controls. What you're seeing, the green here, is just their microglia. They're a type of glial cell. If the area becomes inflamed, these cells increase in their number and they change their size. But this hellish green look over here is an area of a brain on fire, essentially. And so this is weeks after the impact in a rat. That would be years after the impact in a human being. But that's 2 impacts. And so would this be risk for Parkinson's disease as you age if this kind of localized inflammation persists for a long period of time? These are the kind of questions that we're doing in the lab, and many others. And if you've got anything you want to talk about, we're going to be opened up at the end of this to answer your questions. On that note, I'm going to pass over animal imaging to human imaging to Susan. Diana.
Slide, please. Hi, Greg, that was really exciting. And thank you, Dean Hazel Sive, for the wonderful introduction. It's really an honor and a pleasure to be here. Can we have the next slide, please? Dean Sive already mentioned the tremendous issue that we have with the mental health crisis. We had this mental health crisis even before the onset of COVID. And in particular, we've been faced with an epidemic or possibly a pandemic of our teens suffering from severe anxiety and depression. And we may know this from our own family members or students, or by reading the popular press. For instance, the New York Times Magazine recently had an article showing that teens are suffering from severe anxiety, and that these articles are backed up by important science. For instance, to the right here, you'll see that there was a poll indicating that the freshman self-assessment hit an emotional low, a 25-year low recently, where they felt completely overwhelmed. And recently also there was, in 2019, JAMA Pediatrics reported that there was a 100% increase in ER visits with individuals with suicidal attempts. And that really brings home for me, because I have a daughter who suffers from severe asthma, and we were in the ER at Boston Children's Hospital not that long ago. And when we were there overnight, I noticed that there were a number of rooms that had open doors with armed guards. And when I asked the nurse what was happening with those rooms, she said that every room that had an armed guard had a child or a teen who had attempted suicide or had suicidal ideations. And that may have been an off night, but the statistics show that it's not, that actually the suicides have increased 60% in ages 10 to 24 over the last decade. And so this is a phenomenal problem that we face. And we know that there's many issues and challenges in terms of diagnosing mental health illness and in treating mental health illness. For instance, mental health illness is often diagnosed very late. And often, even though there's great patient heterogeneity in treatment response, treatments for mental illness are most often given with trial and error. So usually it's a flip of the coin, or some trial and error basis by which patients who are suffering from mental illness get the prescribed treatments. And finally, a lot of the treatments that are used for mental illness are not fully effective, and they often have very debilitating side effects. Next slide, please. So at NUBIC, which is the Northeastern University Biomedical Imaging Center, which is parallel to Craig's, but we do human imaging, you can see that in the middle here, we have a magnet, and that's an MR facility that we are able to look at brain structure and brain function. So traditionally, we may have used this MRI to look at gray matter structure, for instance, cortical fitness, or perhaps we are using task-based functional magnetic resonance imaging to identify regions of the brain that were localized for a particular cognitive or emotional task. More recently, we've been using this MR machine to look at something called connectomics, which is really to look at how the structural and functional brain network architecture is elucidated by diffusion white matter tracks, as well as resting state connectivity. And today I'll talk about how we have great hope for resting state connectivity in terms of clinical translation. Next slide, please. So here I'm displaying the iconic resting state networks. It turns out that there's ultra slow fluctuations in the fMRI bold signal that exhibit a coherent network in the brain. And it turns out that if you look at these resting state networks displayed here, like the default mode network or central executive, what I'm highlighting here in orange are regions of the brain, even though far away anatomically, they are rising and falling in temporal synchrony during rest. And it turns out that individual variation in these resting state networks offer great insights into brain health and disease. Arguably, next please, arguably the most important or clinically relevant resting state network is something called the default mode network. And I'm displaying the default mode network here in orange and yellow. These are regions of the brain that are more active during self-referential processing or thinking about oneself, typically during which time we're thinking about perhaps reminiscing about the past or planning for the future. And that's when we're in a fairly good frame of mind. However, when we're suffering from mental illness and we're in a negative state of mind, reminiscing about the past may transform into ruminating about the past, or thinking about the future or planning for the future may transform into obsessively worrying about the future. Next, please. And we and others have shown that the default mode network is specifically the medial hubs of the default mode network, namely the medial prefrontal cortex and the posterior cingulate, are more active and more correlated during rest in clinical populations. So people who suffer from psychosis, anxiety, and depression have these regions of the brain that are overemphasized. And the degree to which they're overactivated or overcorrelated corresponds and is associated with more psychopathology. So the more these regions are engaged, the more rumination, stress, paranoid ideation, and other psychopathologies one might exhibit. Next. So the good news about these resting state networks is that they're plastic. We can modulate them with pharmacological and behavioral interventions. So one behavioral intervention that we have invoked is mindfulness meditation. We have provided 6th grade students in the Boston school area 8 weeks of mindfulness meditation exercises. And those mindfulness meditation exercises have significantly modulated their resting state networks like the default mode network and have hence significantly reduced self-perceived stress and increased sustained attention. And what we found is that it was some people had an easier time or a more difficult time learning the mindfulness meditation strategies. And when you learn mindfulness meditation, as well as other behavioral interventions, you have no visual feedback as how you're actually modulating your brain network. So over the course of the years, we've developed another behavioral intervention called RealTime Neurofeedback. And that couples behavioral interventions like mindfulness meditation or behavioral therapy or other behavioral interventions with a mental mirror that allows you to see how you're modulating your brain networks while you're performing these behavioral interventions. So here's what it looks like. We have subjects lying in the scanner. We provide them a mental mirror through which they can see how they're modulating their brain networks while they're performing, in this case, mindfulness meditation. And what we do is we personally localize their particular resting state networks, like the default mode network or central executive network. And then we basically track where the blood flows. So we identify the default mode network and the central executive, and we track how the fMRI signal is flowing over time. And then we convert the fMRI signal into a ball task. And this is the ball task that you see here to the right. So while the subjects are in the scanner, they're also playing this ball task, and they're moving the ball with their mind and their brain. And the way the goal of the task is to move the ball up into the circle above, and the ball moves up when the default mode network is lower than the central executive network. And that happens when the subjects are performing mindfulness meditation, because mindfulness meditation allows subjects to focus and attend to the here and the now, the present, as opposed to rehashing the past or obsessively worrying about the future. And that focus on the present quiet down the default mode network. And because you're attending to the present, your central executive network is increased. So when you're performing mindfulness meditation, your default mode network goes down, your central executive goes up, and the ball moves up towards the circle on top. However, if you start to mind wander, then the default mode network goes up and your central executive goes down and the ball goes down. So essentially you're playing this ball game in the scanner, moving the ball with your mind and your brain while you're watching how your brain networks are being modulated. And this has been incredibly rewarding. We've been able to train patients with schizophrenia who have chronic auditory hallucinations. We've trained them outside of the scanner to do mindfulness meditation. When we put them in the scanner, we train them to modulate their brain networks, to tune their mind to perform mindfulness meditation. And that subsequently modulates their resting state networks. And the degree to which they're modulating, like decreasing the hyperconnectivity, say the default mode network, corresponds to a significant decrease in symptoms like auditory hallucinations. We're extending this intervention to other populations like teenagers with anxiety and depression. We've had the luck of having a large NIH grant where we are following 225 teens who are diagnosed with anxiety and/or depression. And we're bringing them back now for more MRI scans, and we're offering them the opportunity to do this mindfulness meditation neurofeedback intervention. And so far, with the pilot data that we have, we're very excited to show that we can train teens with anxiety and depression how to modulate their brain networks and subsequently feel fewer anxiety and depression and rumination. So thank you. Oh, I'm sorry, there's one more slide here. I wanted to show you an actual video of a patient performing the real-time neurofeedback. Here, you can see the default mode network is in blue. They're starting to do mindfulness meditation here. You can see this patient is suppressing, quieting down the DMN, and the ball is subsequently moving up. The ball moves up into the circle, which shrinks dynamically to make the task more difficult. And you can see the central executive is increasing, the default network is decreasing. This is just one example of many patients who we've successfully used this intervention, and had these patients have subsequently felt significantly reduced symptoms. Thank you.
All right. That is wonderful. What fascinating work, Craig and Sue. Thank you so much for sharing your approaches to brain health, mental health. Really extraordinary way to use brain imaging, looking at things, kind of an old style scientific method of inquiry. You know, science started by looking at things and observing. And here you have taken this into absolutely the 21st century and beyond to really start to address some of the most pressing challenges in human health. I'm looking at our chat and encouraging participants, of which we have a wonderful number, to not to be shy, just put what you want in there, what you want to know. And we are going to now spend some time talking about the challenges of the topics that you're addressing. And I'd like to start by asking you, Sue, to extrapolate for us. You talked about this very interesting approaches of using mindfulness to address depression and anxiety. How do you see that moving forward? You have studies that involve imaging and affected people. How do you see that moving into, let's say, a mainstream treatment that, can be accessed by millions of people? What would be the approaches there?
Thank you, Dean. That's a fantastic question. Thank you, Dean. So we are actually trying to scale this intervention up in multiple ways. First, we're trying to get electrophysiological correlates of basically the brain networks that we're trying to modulate with this intervention. So in order to do that, we have simultaneous EEG and fMRI. So while we're training people to modulate their brain networks in the scanner, we're also passively collecting the EEG. So we can use machine learning and other analytical ways to catch an EEG fingerprint, if you will, at a fast temporal timescale of the brain networks we're modulating in the scanner. And then once we capture that EEG fingerprint, then we can scale that up into real life. And so we're working with industry and figuring out ways we can actually monitor real time and people's EEG and perhaps other electrophysiological correlates like EKG and skin conductance and other things that may be useful so that you have a whole full landscape of information that you can use to then trigger interventions like mindfulness meditation or other interventions. We're also trying to couple this intervention with other top-down and bottom-up interventions. So we are hopefully using this intervention before top-down interventions like cognitive behavioral therapy, because we want to prime the neural circuitry that we want to modulate with cognitive behavioral therapy. We also want to help other bottom-up therapies like pharmacology or like ketamine or other new ways to modulate the brain networks that people are finding useful. And in the case of depression, those particular interventions are really wonderful. However, it's hard to sustain the clinical benefits. So we're trying to couple this technology after things like ketamine to then sustain the clinical benefits, and then perhaps train, once you can use this method to train individuals how to modulate their brain networks with mindfulness meditation, then that could be kind of a bridge to be able to have them carry on this on their own.
Yes, and we could spend actually a long time having you extrapolate, to some little monitor that someone could put on. But I think what, is very interesting about your response is the rigor and the effort and the data collection that is needed before you can actually take your approaches into the sort of treatment landscape that we would all like to see. And I think it highlights the enormous effort that we have to put into scientific research before something that seems so promising can actually get to a place where there is a readout. The scientific effort is absolutely enormous. And although you've made just fantastic strides, the effort still to move this forward into something that would benefit a large set of people, or actually even a smaller set of people, still relies on the ability to perform these rigorous studies with the group of people, your trainees, that you have to do. It's the effort strikes me as just enormous still that you need to put into this.
Very true. I think there is enormous effort. However, I do see a phenomenal opportunity to address some of the key issues and challenges that we're facing in addressing mental health crisis. And the three that I listed initially is that, oftentimes the detection is very late. But if we can use resting state networks, say, for identifying brain-based biomarkers that predict the developmental trajectory of anxiety, depression, and psychosis, then we could perhaps use these interventions very early on before the clinical onset of these mental health crises. And we can also use it to predict treatment outcome, which would be another huge win in terms of clinical translation, so that we can
totally and in no way was I detracting from the power or the efficacy of your studies. I was really thinking out loud for our audience about the enormous effort it takes to do the kind of work that you are doing, and to highlight the absolute effort that goes along with these outstanding strides you're making. Let's look at some other questions here. There is a question for Dr. Ferris. How did you adjust the MRI for the animal's movement? What is the way that you're able to do the very precise imaging that you do in the animals that you're addressing? The data you showed us was just striking in its precision, and it would be important to understand this, and then perhaps to think about this in terms of the human application as well.
Yeah, as I had said, and I could be heard. I'm not on mute. I'm fine. Okay, good. As I had said, if you're being scanned, the only thing that you have to cooperate with is not moving. And so we have to do the same thing with our animals. When I got into imaging, and this has to go back 25 years ago, in a psychiatry department at UMass Medical, all of the studies at the time were being done on anesthetized animals. And I just would, it should be obvious to all of you, you're not going to learn much about the brain if it's anesthetized in terms of its activity. So I literally started a company by writing grants to the National Institute of Health to start a company to make the technology and electronics and holders to do awake animal imaging. And so there's an entrepreneurial point in my life years ago to get the tools I needed to do my science. And obviously I love academia, so never left academia. So the technology is out there for taking any animal, and that would be, and we study voles and mice and rats and hamsters and small monkeys and ferrets, and we study them while they're fully awake. Now, the only thing that actually has to be held stable is the head. The rest of the body can do whatever it wants. And so we have technology where the animal can move its body and be comfortable, but its head doesn't move, and it's all passive restraining systems. So, I noticed that there were a lot of students and other faculty and other people associated with Northeastern there. If you want to come in and see what we do and how we do it, just pop by the CTNI. I hope that answered your question.
Very interesting. There are some wonderful questions here. Craig, I'm going to give you another one because I see someone is asking, Nina Miller's asking about excessive microglial activation, as in Brain on Fire. And I don't know, Nina, if you mean that there was a wonderful book called Brain on Fire that I thought was really fascinating. So, what about the connection between neurodegenerative diseases and these initial insults to the brain through brain injury? Do you have insights from your research right now?
From our research, what we find is, and we've published actually now several papers where you can insult the head in the front or the back of the head, and there are common areas across globally over the brain of our rodents, certainly, that manifest with an increase in neuroinflammation. And this could be with different biomarkers. And so the biomarker you were looking at was, IDA2 or IDA1. So it's a biomarker for activated microglia. We study GFAP, which is another biomarker for astrocytes. And so you can do immunohistochemistry and see the change in these biomarkers, which are just, a measure of neuroinflammation. What has been so surprising to us is how long these things persist after these kind of insults that are not cracking the skull. There are no what would be called a contusion, where there might be a rent in tissue and blood vessels break. There's none of these things. These are really mild head impacts, but have long-term consequences. So I think underlying neuroinflammation is a serious problem with any brain injury and how long it persists. And probably therapeutics should be addressed in this area.
Thank you, Craig. I was actually going to follow that up by saying, have you or others done pilot studies to try to use anti-inflammatory treatment that would get across the blood-brain barrier and see whether or not that clears the inflammation quicker and changes? You'd have to do a long-term study on that. That seems to be your data really suggests that could be a very useful route.
Yeah, One of the advantages, and I would also call it a disadvantage in some respects, one of the advantages to the animal imaging is that we can do prospective studies. We could take a normal, healthy animal. This is something you can't do. Susan can't do this. She can't have her kids come into the lab and bop them on the head and say, okay, come back in another week and let's see what's going on. So these are the advantages, certainly, of animal imaging. We have more experimental latitude, as we call it. So we can bring a normal, healthy brain and we can do these things to it, and we can watch it over its life. Now, its life is 2.5 years. And so these kind of studies where you're doing these aging studies to early risk are very expensive, okay? And not one that's normally and easily funded by NIH. It's just like, okay, we get it, and it's probably a serious problem, but has anybody ever demonstrated that you take an environmental factor, you take a genetic risk factor, and you combine them with aging? For instance, Parkinson's. Parkinson's, we believe that this is a risk factor for Parkinson's. That's true. It's been recognized in the health community. Head injury of this type is a risk factor for Parkinson's with aging. That combined with all of the genetic risk factors for Parkinson would really put somebody on a path of no return, so to speak, as soon as they lose enough dopamine neurons. These kind of studies are, longitudinal, long-term studies that are not easy to get funded. It's like, tell us something that we really kind of know, but no one's fully demonstrated. So the point is, and that Hazel's making here, so well, if you go in with a really good intervention strategy, wouldn't that make a lot more sense in these long-term studies? And that's a good one. So the question is, what would be that intervention strategies? And this is certainly something that's not been lost on big pharma and biotech. And so, but they have their own agenda. And what would be that agenda then for the National Institutes of Health? What drug would they pull out and say, this is worth doing, that a drug company is not already doing, that's already in clinical trials? So it's kind of an interesting, great question, kind of a dilemma from a preclinical academic approach.
That's very interesting. That is indeed points to the complexity of doing this groundbreaking research that suggests things, and you then have to figure out how to actually take it to fruition. And that is, again, a huge amount of effort. Let us go to a couple of questions for Sue. Someone asks, Sophia Baldino asks, if you've observed instances during which patients are unable to suppress the DMN, leading to something similar to a negative feedback loop, where patients find it increasingly more difficult to initiate mindfulness meditation?
That's a fantastic question. And actually, we haven't actually seen that so far. People fit to the zone, so to speak, where they're able, because they're kind of dual tasking with the ball task and doing mindfulness meditation. So we haven't seen the negative cycle or closed loop where they're actually getting frustrated so that they actually have less ability to do mindfulness meditation. Rather, we do see some people who are less able to do it initially, but then they catch on. And so it was really the mindfulness meditation that has really been able to help us modulate these networks more than any other kind of dual task intervention. We've given many, many different strategies, and some worked and some didn't, but the mindfulness meditation, people across all different kinds of clinical populations seem to be able to get it. And we actually haven't had a negative feedback loop so far. So that's been a positive experience with the mindfulness meditation.
That's very interesting indeed. Jude Matthews asks, will mindfulness meditation help in the management of neuromuscular disease, do you think?
I think absolutely it could. We have we started this research many, many years ago back at Stanford, where we trained people to modulate their motor cortex. And then after that, we took individuals who were suffering from chronic pain, and we were able to help them modulate their anterior cingulate that subsequently had them experience reduced pain. So I haven't actually looked at neuromuscular disease per se, but I think this application could be extremely beneficial for that particular target.
And here's a different related something, a couple of other questions here for you, Sue. Have you encountered a difference in patients who are suffering from depression or anxiety and are on medication versus not on medication when doing the mindfulness training? So is there a synergy there? Is this a substitute for medication? What does your data tell us so far?
That's a brilliant question. And unfortunately, I don't have enough data to actually answer that question, but we do have these 225 individuals who have been diagnosed with anxiety and depression. And we are just now, we have 10 so far, but some of them are on pharmacology, some of them are doing alternative behavioral interventions like cognitive behavioral therapy or DBT. And so we could actually answer that question, hopefully in the not too distant future. But right now, we don't have enough data to answer. And I'm hoping that this intervention could augment the current treatments and possibly even replace them, especially because we can now offer this behavioral intervention for children who may be at genetic or clinical risk for developing a disorder, but who are not yet ill. I guess the short answer is we don't know yet, but we are hoping to find out soon.
And as I understand it, Sue, the real key to your procedures is that you're using imaging together with the mindfulness meditation so that there is a feedback that you can put together. It's not just, some commercial company, you see on TV, mindfulness meditation, whenever you like, just call us up and pay your monthly fee. But what you're doing is to actually take this kind of lifestyle and then couple it with this very high end imaging and feedback connectivity so that you're going somewhere from something that's sort of popular to something that is scientifically modified. Is that a correct?
That's exactly spot on, Dean. So thank you. So yes, we are trying to use this as the MR machine is providing a mental mirror to see how you're modulating your brain networks during mindfulness meditation. This could be applicable for other behavioral interventions like emotion regulation and so on and so forth that has been shown to then improve treatment efficacy in subsequent interventions. And also we've been able to use the real time in a different context. It actually changes human performance. So we basically can track when a brain is in what we call an optimal brain state or a poor brain state. So you can have an optimal brain state for memory. And so we can track the brain signatures and trigger a particular task, say a scene. And then later on, we can have the people respond whether they remember that scene. And we find that people are actually having better memory for the scenes that were triggered when we have identified a good brain state or a bad brain state. So in the future, it would be really interesting if we could take EEG in combination with other measures to basically do an online tracking of a person's body or brain state that then would be predictive of a subsequent symptom, and then basically trigger an intervention like mindfulness meditation that you can do by yourself that then would modulate these brain networks. So there's a couple of different directions that you can imagine going, but you're correct that unlike many of the Headspace and other mindfulness meditation apps that you have out there, this particular intervention allows you to see how you're modulating your brain networks while doing the mindfulness meditation, which will then in turn allow you to learn it, accelerate your ability to modulate these networks, and then that would in turn improve the clinical and cognitive benefits.
Thank you. Very, very interesting. I'd like to take these last few minutes to ask each of you for a projection. If you could think of something that would give you really a stunning projection into the future. We've titled this panel discussion, The Plastic Brain, and both of you have indicated through imaging how the brain is plastic after insult, after injury, and during recovery from anxiety and depression. Could each of you speak for a couple of minutes about what you see as an extraordinary direction to take each of your research as you go forward and to address brain plasticity in the most positive sense? Craig, perhaps we could start with you, and then Sue, you could give us a bit of a wrap up.
will give you an extreme example. We published a paper 2 years ago, and the paper was entitled Life Without a Brain, Rat 222. And in our imaging of a 2-year-old rat, which is a very old rat, we discovered that the animal literally had severe hydrocephalus, and most of its brain was gone. Okay, it had no visual cortex, it had no auditory cortex, its somatosensory cortex was gone, its hippocampus disappeared from its normal site. So when we recognized we had a unique animal from imaging, we ran a number of studies on this animal. We tested it for visual acuity, we tested it for cognitive behavior, we tested it for hearing and sensitivity. All of these different sensory modalities were normal. Okay, we couldn't identify this animal from any other, and it really speaks to the plasticity of the brain and our hubris about how complex our cortex is, how important it is. But on that note, okay, there are ways of looking at brain plasticity and understanding that nature, okay, has solved a lot of these problems. And when we do our research, we tend to focus on how would nature, how would the biology of evolution deal with this issue and with this problem. Evolution says you can get hit on the head once and you should be perfectly fine. Why would they ever design you to get hit on the way, and then you're out of the gene pool? Okay, but you got to be pretty foolish to keep going back to the same circumstance and getting bopped on the head over and over and over again. All right, so nature's behind the scene helping us get through the life that we have. All right, at some time in there. And the beauty of imaging is that you can follow changes in anatomy and function in the same animal over the course of its life. And it's totally non-invasive, and it's incredibly exciting and a lot of fun.
Thank you. It's a great story. As someone who has hydrocephalic fish in my lab, I think that is a great story.
You got to image your fish.
Totally. Totally. The audience doesn't know, but this topic is very close to my heart. My own research group uses zebrafish to understand something about human mental health disorders. Sue, perhaps you could give us your projection as to what is exciting, what is somewhere that you think about particularly and say, wow.
Well, there's two directions. One is that there's a behavioral version of something that was recently published in Nature Medicine a few days ago, where that particular paper illustrated that you can use a closed loop neuromodulation in a single patient with depression using deep brain stimulation. So they identified symptoms for that particular person, and then they were able to use deep brain stimulation to perturb the brain system that then had subsequent reduction of symptoms in that particular individual. And I'm hoping I could see a time where we could actually do a behavioral version of that, where we could do longitudinal assessments, identify symptom or state specific constructs using MRI, and then eventually electrophysiological measures that identify the brain behavior relationship. And then with a closed loop neuromodulation technique, we could, on an individual symptom specific basis, be able to modulate those symptoms over time. And in terms of the brain plasticity, if you look over the course of the lifespan, you can think that there might be critical windows for which you can perturb the brain, and especially for disease-specific windows. For instance, ADHD or autism may be a little bit younger, psychosis a little bit later, so on and so forth. So if we could identify by using wearables when a person might be more vulnerable to slip into a negative symptom state, then you might be able to invoke this kind of closed loop behavioral neuromodulation at a very young age, so that could mitigate symptom progression and possibly even avert clinical onset.
Thank you. That is a very exciting projection indeed. I want to thank you all for being here today. Again, I want to thank our speakers, Craig Ferris and Sue Whitfield-Gabrieli, for sharing their insight, sharing their research with you, for letting us think together about some of the ways that imaging and the plastic brain together are helping move forward the future of addressing brain injuries and the devastating impacts, and the future of mental health disorders and their devastating impacts. It's an honor to have you both as colleagues, and it's an honor for us as your audience to have heard about your work today. Thank you so much, and thank you to you all for being together with us and learning about some of the extraordinary work that is going on in the Northeastern College of Science.